let's go ahead and get started. Before we dive deeply into the biology of mind, the very exciting neurology and brain stuff. Remember last time I was talking about how my livelihood and the grad students' livelihoods depend on research? We have one of our grad students here to make a plea for help with data collection for her master's thesis, so she can finish on time here. So I'm going to hand it over to Christina.
Yes, so hello everyone, my name is Christina and I'm a second year grad student. I'm trying to defend my thesis this semester and trying to graduate, hopefully in April. So I'm kind of like getting last set of data collection today, Friday and next week. This is my information. If you have any questions, that's my email and that's the name of the study I'm conducting. It's up on Sona and it has two parts. The first part is online, second part shouldn't take more than 45 minutes. You will get 3 activity points, which I know you need, and you will really help me out. So if you have any questions, anything about signing up, just send me an email to the email address. And that's all I have. I hope a lot of you sign up if you haven't done it yet.
Any questions for Christina?
So on Sona, what's the study called?
It's problem solving, emotional perception, and personal attitudes.
You see it?
And it should have part 1 and part 2.
Yes, ma'am.
So it says that there's no time slots currently available, so does that just mean it's full?
That is
Are you having the same thing?
weird. Okay, so when I was doing this data collection, some people were having trouble like signing up, and apparently this student here, it says that there is no more available times, which is where it might be that you can't really sign up for this week, but you will definitely be able to sign up for next week. So if you're having trouble, just email me because I can actually sign you up manually, and that's what I've been doing with some students. So just email me and I'll do it, and then I'll send you an email saying that you've been signed up. I'll send you the time slots so you can tell me which time fits best for you, so I can actually sign you up.
Any other questions? Awesome. Thank you, ma'am.
Thank you. Nick.
Don't forget your phone. All right. So today, talking about the biology of the mind, how it works, what are the individual components that make it work. We're going to get down to the individual neuron level, and even smaller than that, the neurotransmitters. Are you really excited about that, or are you excited about something else? Just lie and say this. Yeah, okay, perfect. The individual neurotransmitter level, which is what's communicating between neurons, and then we'll take it from there. This is a side note, has something to do with this class. But if you haven't watched it yet, there's a 3-part documentary on Netflix about Aaron Hernandez, former Patriots tight end, who was accused of murder and wound up committing suicide in prison. But the last part of the documentary is all about CTE. So and that is what a lot of football players seem to be experiencing now as a function of repeated head trauma. And I talk about the brain. And there's some really interesting cross-sections. So after he passed, his family was willing to donate his brain to, I don't remember what institution it was, maybe New York, for them to do a study on his brain, and they've got cross-sections of his brain, and you can start to see some of the actual structures we're talking about in this class. And then also, what are some of the consequences of, for example, repeated head trauma. So that's just an aside. It's a really interesting documentary, and you get to work neuroscience into it. So, extra exciting way to spend your weekend. What did we learn last time? Yeah. I'm sorry?
Twin studies?
Yeah, twin studies. What are twin studies? Okay, so yeah. So we're studying twins. And why are we studying twins? Somebody else. No, go ahead.
A lot of like nature versus nurture can be answered them if they're separated.
Yeah, it allows us to establish, okay, how much of a trait or behavior or manifestation of certain genes is attributable to nurture, so your environment, and how much of it is specifically nature, so your genes. What else do we talk about? Yeah.
I watched that video on mice where they were like exposed to a smell and they were shocked, and then they found that when they separated the kids of those mice, that they had the same reaction when they were exposed to the shock.
Yeah, so we watched a video where they exposed some mice to a smell and then shocked the mouse. So they created this association between the smell and the shock. And then they took the offspring of those mice, separated them from the parents, so there couldn't be any sort of environmental influence on these mice. And they still saw a response to that smell in the baby mice. And so what do we call that focus in science? Yeah. Heritability or
Epigenetics.
epigenetics. Yes, exactly. So again, this idea that you can have essentially genetic memory. Right? And so after class, a student came up and said, my dad's really afraid of heights. I'm really afraid of heights. Is that something I inherited? Or is that something I learned? And so I think the answer is probably a little bit of both. Right? And so if you see your parents really afraid of something, chances are you're going to be really afraid of it. Right? As you develop and you learn this sort of fear, the association of fear, you're going to develop that as well. But there also may be a genetic component. So maybe at some point in the lineage of this student's ancestry, there could have been something that triggered a kind of catastrophic fear of heights. And that gets passed on to subsequent generations. Now I maintain that a fear of heights is not actually a phobia, because phobias are supposed to be irrational fears, and heights are very irrational fears, very legitimate. Maybe that's just my way of convincing myself I'm not a total wuss, but I too am deathly afraid of heights. What else are we talking about? Yeah? Temperament? Okay, in what context do we talk about temperament?
If it's, like, for example, if it's through, if we're studying, like, identical twins, do they have, like, the same, I guess, emotional reaction to things?
Yeah, exactly. So how much of who you are, your temperament, how excitable you are, is due to genetics versus environment, right? And we all like to think we have complete control over our emotions, but in reality, there's a substantial part of our emotional temperament that is attributable to genetics. What else? Maybe one more thing we talked about last time. The marshmallow experiment. What is the marshmallow experiment? You gotta talk loud enough for them to hear. I don't want to keep yelling.
They stick kids in a room with a marshmallow and they tell them if they wait, they get 2, and if they eat it now, they get to eat it.
Yeah. Right? So they took kids, they put them in front of a marshmallow. The experimenter said, I'll be back in 5 minutes. You can eat the marshmallow if you want, but if you wait those 5 minutes, I'll come back with a second marshmallow, and you can have 2 marshmallows. And so they did that test, and then they studied those kids over time, and they found that those who had the ability to wait, delayed gratification is what we call it, tended to be more successful throughout life than did those who ate that marshmallow right off the bat. Now, does that mean if you ate the marshmallow right off the bat, you weren't going to be successful? No, of course not, right? Nothing we're talking about is an absolute perfect correlation in that it predicts all the variance. Right? This is just saying that if you waited for that second marshmallow, you're slightly more likely to be successful later in life. Any questions about what we covered last time? Points of clarification? Awesome. So, you have an exam coming up soon. What questions at this point do y'all have about the exam? Yeah?
Could you tell us how
It will almost certainly be all multiple choice, true, false, and then like a fill in the blank.
How many questions?
I'll answer that question, but first you have to tell me, what does that information do for your ability to study?
Well, my next question was, do we get the whole block, or the whole time period to take it?
Yes, you'll get the whole time period.
Okay, the questions don't matter.
Yeah, there you go.
Is it based off your notes and the book, or just one or the
Both. Is that a hand? Yeah.
So when you structure your test, is it more like, is it so further questions, the ones like the ones that are looking at, what is it, more application based or like definition based? Like what is it?
So there'll be a little bit of both. It won't be definitional just for the sake of being definitional. It'll be definitional to make sure you're able to use a term and understand what it means, if that makes sense. And because of my snafu, with us pushing everything up now, we're going to have a full devoted review session. But the caveat for the review session is you have to have studied prior. You have to come in with specific questions. You can't say, can we talk about that whole biology of mind lecture again? It's not going to cut it. And remember, all of these lectures are recorded, and a link to the recorded lectures are posted under the announcements tab on Canvas, so you can go back and suffer through listening to the lectures again. Any other questions? Cool. And then somebody who listens to the lectures provide me feedback about how audible my voice is. I hate listening to myself talk, and so I refuse to actually play through it to make sure everything's recording the way it is. So if things are messed up, let me know, because every time I go to download it, it starts auto-playing, and I rush to hit pause because it's like nails on the chalkboard. So that's my own insecurity, but you're going to have to deal with it. All right, here's your fun fact of the day. Ravens in captivity can actually learn to talk better than can parrots. Second fun fact of the day, this isn't up there, but Andrew Jackson, President Andrew Jackson, had a parrot, and Andrew Jackson was known for having a salty mouth, swore a lot. His parrot was actually removed from his funeral because his parrot kept swearing at the funeral. So, there you go. Yeah?
I saw a news article. It was firefighters were trying to rescue a parrot. It was trapped. And it kept telling them to fuck off.
Yeah, you have to wonder what the environment is that leads parrots to essentially just parrot that sort of bad language. All right, so at the very end of last class, we were talking about how early experiences can affect your development and can have long-lasting effects on brain health, brain development, etc. We talked about things like malnourishment, but also abuse, and not even being necessarily the subject of abuse, but witnessing abuse. One of the reasons there is such a dramatic impact early in life is because of what we call synaptogenesis. So when you're born, and in the first 2 to 3 years of development, you reach the peak number of synapses that you're going to have in your brain. You're not going to ever have more than that, and everything from there is downhill. So yes, you peaked at about 24, 30 months. It's preparing you for learning and development, right? It's getting the whole playground ready, so to speak, for you to use as needed in the development throughout life. And this peak is about 15,000 synapses per neuron. So for each individual neuronal cell in your brain, there are 15,000 connections to different neurons. That is unbelievable complexity. However, after that peak, what we call pruning happens. And pruning is the essentially subtraction of these synapses that are not being used. And that's why it's so critically important in the first 10 years of a kid's life to engage those synapses to make sure that they're not losing those during this pruning process. And so the whole adage of if you don't use it, you lose it, that absolutely rings true with synaptic pruning. This is also one of the reasons, this brain development, all the way up through adolescence, why kids need so much sleep. And why y'all, I mean, you're kind of on the tail end of it, but why y'all need more sleep than regular adults, because it is inherently exhausting to the system to go through this process of initially building up these synapses in the first 2 to 3 years, and then paring them down. That's why I think last time I saw, suggestion was that teenagers should be sleeping about an average of 10 hours a night. Yeah, some of y'all are laughing, I know. And so we talked about the effects last time, just generically, of trauma on the development of children. And unfortunately, there are various case studies. And again, what's a case study? We talked about this before. Yeah, it's just a single individual that we really drill down into their history to get all sorts of information. There are a lot of these case studies of horrible neglect and abuse that allow us to understand the consequences of this sort of isolation. And so there was a young girl, given a pseudonym Jeannie, who was found locked up in her parents' house and had been locked up for about 13 years. She was almost always strapped either to a child's toilet or bound in her crib. There was no sort of stimulation, no sort of external interactions. After being rescued, I mean, this was kind of, ripe for scientific discovery, in addition to the benefits of liberating this child from these horrible circumstances. The question was, okay, at 13 years old, how much of these functions can she regain? Right? I mean, at the point she was found, she had virtually no social function, no language abilities, almost no motor skills whatsoever, because she had been bound in a way that didn't allow her to interact with her environment. And after this point, she was able to regain slight functions, slight social functioning, ability for crude language use, and gross and fine motor skills, but it never developed to the degree you would see a normal developing child to develop. And again, because at 13, you've already had this substantial neural pruning, so your brain says, okay, well, I'm not using that pathway, get rid of it, use that for something else. And so the ability to redevelop skills is not as readily available as if she had had the normal development and normal stimulation as a child. And so the ability to learn some of these functions, even after she was recovered at the age of 13, is representative of what we call plasticity in the brain. So the brain can sometimes change to compensate for lost functions with damage, for example. So if you've had an elderly parent or a grandparent who's had a stroke, there's an initial loss of function. And after a stroke, that part of the brain dies. And so there's not the ability to essentially reuse that part of the brain. So other parts of the brain have to learn how to compensate to restore various functions. Sometimes there's no restoration of the functions, but frequently the brain can at least in part restore these functions. They can also sometimes adapt to new experiences and development itself. That's why you can learn as an adult these skills, but it's better off as a child or an adolescent before you've had this full neural pruning to actually learn these skills because those synapses stick around. Yes, ma'am?
I saw a study about this guy who couldn't talk, but there's a different part of the brain that allows you to sing, and so he could train himself to sing in order to like talk. Is that like an example of plasticity, like the brain being able to find other ways?
That's really interesting. So, not exactly. If he had used that region of the brain that was for singing to learn how to talk, that would be an example of plasticity. This was kind of learning to adapt to that. Presumably he never learned to talk in this case. I don't know this case specifically. But learning to sing is just using the part of the brain that would be responsible for the singing in an adaptive way. So it's not exactly an example of plasticity, but it's a really interesting demonstration of kind of the ways to overcome some of these deficits. But great question. So I'm going to show you a really neat example of plasticity here. First I'm going to increase the volume.
I'm Daniel Fish, and I'm the president of World Access for the Blind. I lost my first eye at the age of 7 months and my second eye at the age of 13 months. Echolocation is actually very simple. The physics of echolocation is very simple and easy to understand. It's similar to vision in a lot of ways. You have energy, in this case acoustic energy instead of light energy, and the energy goes out from the individual. In our case, it's in the form of a tongue click. And then that energy bounces off of everything in the environment, all surfaces everywhere, and comes back to the listener. So what I'm going to do is I'm going to familiarize myself with the area by clicking and listening to the returning echoes, and also keeping track of where I go and where I come from. Now there's a structure here, a small one, or well, it's a pole. I see. So now it really does open up. Here's another pole. And this is probably a court. Yeah. So this would be a basketball court. And there would be the hoop. For us, it's all about freedom.
Right? It's incredible to think about the fact that we as humans can develop the ability to echolocate, right? We think about bats as being, unique in some capacity. In the second half of this video, I'm not going to show it, but he teaches a novice essentially to go through and develop this ability for echolocation. So what's likely happened in this case, you heard him talk about the fact that he lost his sight at a very young age. He's probably incorporated the visual parts of his brain, this occipital lobe here, into processing information from his auditory system. So he's likely rewired his brain at that early age to incorporate those visual areas, or what are typically visual areas, for processing of the information that is normally processed here in the temporal lobe, across the temporal lobe, and into the occipital lobe as well. So again, you can see this as a representation of what the brain's able to do if it has kind of a blank slate, so to speak. Right? It has all this computing power. It has all this processing ability. And what it's doing is looking for inputs that are stimulating the parts of the brain. Parts of the brain that get stimulated stick around. Those that don't get stimulated get tossed to the wayside. And so in this case, because he had an entire region of his brain devoted just to vision, and yet had no visual input, his brain was able to reincorporate this sort of visual processing to make sense of his world in a non-visual way. Here's another example. I'm not going to show the full video. This is fascinating. So, as a preface here, they engineered a bike. So, does anybody not know the Smarter Everyday YouTube channel? Okay, good. For the couple that raised their hands, it's an awesome channel. He actually lives here in Huntsville. But what they did is they engineered a bike to steer backwards. So when they turned the handlebars to the left, the wheel went to the right, and vice versa. And he talks about the unbelievable struggles of when he first started trying to ride it. I'm going to skip
Hey, it's me, Destin. Welcome back to Smart
parts here. You don't need to actually listen to him talk. But here's the first time he attempts to ride it.
I couldn't do it. You can see that I'm laughing, but I'm actually really frustrated. In this moment, I had a really deep revelation. My thinking was in a rut.
So he then talks about how he takes this bike to different speaking engagements because he says nobody believes him that it's actually this hard. And he offers $200 to anybody that can ride it 10 feet. And doesn't get anybody who can actually ride it 10 feet. And what you'll see, I don't know if this is one of them, but people will like just hop along the ground completely incapable of actually riding the bike fully here. Let's give another example. This is my favorite. This guy refuses to lift the foot up off the ground and keeps going as if he's doing it. Much to Dustin's chagrin. So, now that you've seen that...
My neighbors made fun of me. I had many wrecks, but after 8 months, this happened. One day I couldn't ride a bike, and the next day I could.
So it took him 8 months to learn how to remap his brain to ride the bike.
It's really weird though. It's like there's this trail in my brain. But if I wasn't...
But one of the amazing things is he did the same thing for his son.
That's over half his life. I wanted to know how long it would take him to learn.
That's a normal bike.
So I told him, if he learned how to ride a backwards bike, he could go with it Australia. Are you going to give up? Go ahead. This is how it starts. Look at this. This is such a big deal.
So he was able to do that after... he ate it. He was able to do that after 2 weeks of practice. Right? So the idea is his brain had a lot more of these synapses to incorporate, hadn't gotten stuck into this rut of thinking of the process by which you have to ride the bike, and was able to ride a lot further in those 2 weeks than Destin was able to after months and months of training. Now the additionally interesting thing is he then records this video of him in Amsterdam trying to ride a regular bike after learning to ride this backwards bike. And he's completely forgotten how to ride the normal bike.
So you can't learn both.
Maybe with enough practice when we talk about things like expertise, maybe.
At this point, I'm pretty sure that all I proved is that I can only redesignate that bikes.
But yeah. So what he's talking about there is he essentially just redesignated the parts of his brain that were understanding how to ride that backwards bike, or excuse me, learning how to ride a regular bike into learning how to ride that backwards bike, and he didn't have the ability to maintain kind of simultaneously this ability with both bikes. And then you'll see something here. This is after about 20 minutes, and all of a sudden it then just clicks for him, and he's able to start riding again. And he talks about all these people who are watching him do it who didn't believe him that he actually didn't know how to ride a bike, right? Because it's an inherently unusual thing to see of somebody who can't ride a bike for 20 minutes and then all of a sudden can start riding a bike. So he had to redevote those neural pathways to learning how to do this. Because this was a skill that he had for the longest amount of time, it was a quicker acquisition of those skills. But you can see how using the brain, there was not the ability, the plasticity that his son has to learn these new skills, but was able to learn how to do it by simply devoting the resources he had from learning initially how to ride a bike. I thought about it too late, but I almost wanted to reach out to him and see if we could bring the bike in here to get one of y'all to try to ride it across.
If one of us could ride it 10 feet, will give us an A.
Bonus points?
Sure. If you can get the bike in here and ride it 10 feet without ever having practiced before, yes, I'll give you an A. But it has to be in the next 30 minutes. All right. So, we've talked about neurons to this point. We haven't actually defined what a neuron is. So, the neuron itself is represented here and has a handful of different components. And so we'll talk about the actual functions here in a second, but the dendrite is receiving the message, it's transmitting it through the cell body. Right here, this little triangle area, is what we call the axon hillock. And when we talk about how these neurons work, in just a few minutes, you'll see that this is where the actual electrical signal originates from. We then have, under these yellow blobs, what we call the axon itself. Wrapped around the axon is what we call myelin sheath. Does anybody know what the purpose of myelin sheath are? Conduction, yeah. It's essentially serving as electrical tape on your neuron. And if you've heard of, for example, multiple sclerosis, MS, it's just a degradation of the myelin sheath. Easy to remember, myelin sheath, MS, MS, MS. And then, I don't know what the heck happened here with the letters, but then you have the axon terminal, which is what's releasing the chemicals that's picked up by the next neuron's dendrites there. And so we're going to break this up, talk about each one of the components individually. And so the cell body, the soma is what we call it, right in there, is responsible for providing energy and the proteins necessary for repair and generation of new parts. It also has the certain components that are necessary to make the new replacement parts for the neuron. And then also has the components necessary to assemble the parts into the neuron or to fix the neuron. So you can think of the cell body as essentially the mechanic shop, right? You go there, it's got all the parts you need. If it doesn't, it's got the tools necessary to make the parts, and then it puts the parts together. The dendrites, as we show up here, are what receive the message from the other neurons via what we call neurotransmitters. We'll go over some examples of the neurotransmitters here shortly. But they then take this information in the form of neurotransmitters, which are just chemicals, and transmit it to the cell body via an electrical conduction. So again, you're taking one source of information, chemical, and it's translating it into electrical. The axon itself, I really should have put in this slide every other page, is this long stretch right in there underneath the myelin sheath. And so after you get this conduction of electricity, it travels down the axon hillock right at the base of the soma and down the axon itself. This sort of electrical impulse is what we call an action potential. I don't know where the name comes from, because it's not a potential action, it's an actual action. But one of the cool things about these action potentials is it exists as an all or none state, right? It's essentially, think of it as a binary state. It fires or it doesn't. It fires or it doesn't. There's no graded action potential. It's not like half of an action potential. It either all fires, that one neuron, or it doesn't. To fire, it has to reach this certain threshold, right? This is the point at which it runs away and continues on with the firing process, and we'll talk about the mechanism of that in just a second. But you have a certain stimulus, right? You have an input from the presynaptic neuron. And you may have a little activation here, but it doesn't reach that threshold, and so nothing's sent. Little activation, doesn't reach the threshold, nothing's sent. Once it finally reaches that threshold, boom. It polarizes, becomes more positive. And again, we're going to talk about the mechanism, but you have what we call sodium and potassium pumps, right? And so it's pumping sodium ions into the cell and pumping potassium ions out of the cell. And this is what's creating that action potential. After the neuron itself fires, we have this period of hyperpolarization, which is also what we call a refractory period. So in this refractory period, it can't fire again. It doesn't matter how many strong signals it gets, it can't fire again until it reaches this resting state once more. And you can see the relative duration, or actually the absolute duration, of the firing here down on the x-axis in milliseconds. And so you can see the whole process from stimulus to resting state is about 5 milliseconds. Right? So everything we're going to talk about here, of the actual firing, the sodium and potassium pumps, everything's happening within about 5 milliseconds, then ready to fire again. And when we talk about the visual system later in the semester, we're going to show an example of single cell recordings, single neuron recordings in a cat's visual cortex, and you can hear an auditory representation of how quickly it's firing. So again, you've got these sodium and potassium pumps here, and you've got a ratio of travel here, right? And so for every 3 sodium ions that are pumped into the cell, right? So this is outside the cell, this is inside the cell. For every 3 sodium ions, 2 potassium are pumped out. And that's what's creating this change from negative 70 millivolts, which is that resting state for the neuron, to a positive state, where now we've got a positive charge inside the neuron compared to outside the neuron. You reach a certain point, and then you have continued pumping of the sodium and then also potassium ions outside of the cell, which is what's creating this hyperpolarization and then the leveling off. Questions about this so far? So, calling it a pump is somewhat misleading. I mean, there is an active process, but the mere fact that you have this differential in charge from the inside to the outside of the cell is making it such that as a passive process, all it has to do is open the doors and you get an equilibrium that generates across that cell. So again, remember, everything that's happening here is happening in about 5 milliseconds. 4 to 5 milliseconds. And so that membrane must be depolarized beyond that threshold. Right? We showed on that first graph what that threshold is. Anything that is not reaching that threshold is not allowing that neuron to fire with that action potential. As we just talked about, that depolarization opens voltage-gated sodium channels, which is allowing that sodium to flow into the actual neuron itself. At the peak of its firing, of that action potential, those sodium channels close, the door closes on it. However, the potassium channels stay open, and potassium ions are still pushed out of the cell itself. However, it kind of overcorrects, so to speak, in that it continues to pump out a lot of this potassium, these potassium ions, such that you get this refractory period, this hyperpolarization, leading to the neuron not being able to fire. Those pumps are then, as more of an active process, creating that resting state difference again, with more sodium on the outside, more potassium on the inside, so it can then fire again. Again, this is amazing. All of that's happening in 4 to 5 milliseconds. For those of you all who don't remember metric, there are 1,000 milliseconds in a second. So essentially in 1/20th of a second, it's firing. Excuse me, 1/200th of a second. So I think it helps to have visual imagery when thinking about some of these kind of more abstract concepts. I mean, the concrete concepts, but it's hard to actually see. And so if you've ever been to a water park, you have these little tilty buckets, right? They fill up with water, they reach a certain point, and they dump it all out. Correct? They fill up with water, they dump it all out. What you don't have is these buckets sometimes pouring out a little, sometimes pouring out a lot. It's pretty much the exact same every time. It pours out, then you have this refractory period where you have to wait before it can then pour out again. It reaches this tipping point. And that's exactly how we should conceptualize these action potentials as an all or none action. So again, going to the myelin sheath, it essentially helps the speed of conductivity down the axon. It's helping your brain process faster by essentially wrapping this electrical tape around the axon itself. I mean, this is one of the things that always strikes me as unbelievably cool about the brain. Right? Not only does the brain have the ability to receive messages in the form of neurotransmitters, it then translates those messages into an electrical impulse. That electrical impulse then travels down the axon to eventually release more neurotransmitters, and again, we'll go over that in a minute. And as if that weren't amazing, evolution got us to a point where we've got this essentially benefit, this heightened ability to transmit the message even faster. We've got cells in the form of these myelin sheath whose sole purpose is to allow us to communicate faster. It's a hyperspeed internet connection from one neuron to the next. And it's just normal. It's unbelievable. And so terminal branches are what exist at the end of this axon and what are ready to transmit the message to the next neuron. You also have neurons that directly innervate on the muscles, and it's what sends signals to your muscles to contract, etc. Do I have a picture? No. All right, so here's what we're going to do. We're going to create a new slide here. All right. Let's see if this works. My first time actually trying to use the pen here. There we go. All right. So you have up here what we call the terminal branch. And within the terminal branch are these little pockets of the neurotransmitters. We call this the presynaptic neuron. And then on the neuron receiving the input, what do we call this down here? Dendrite. Yep. So we have the dendrite ready to receive the information via neurotransmitters from the presynaptic neuron. We call this the postsynaptic neuron. And we call this, call this area right here the synapse. That's where the presynaptic, postsynaptic comes from. And what you have on this postsynaptic neuron are little, essentially garages, for lack of a better term. I don't know why I'm thinking all auto today, but garage, for the neurotransmitters to come in. And each one of these receptors right here has a specific design or a specific shape. And so to oversimplify, we'll say we have 3 different neurotransmitters up here with 3 different molecular shapes. These receptors down here have the ability to receive only one, sometimes more than one, but for this case, only one type of neurotransmitter. And so you may have this, we'll call it serotonin, come here. It's not able to bind there, kind of hangs out in the synapse, and then we find that it can bind there, creates this sort of activation. Same thing, we'll call this, dopamine, comes down, directly binds. Epinephrine in there, directly binds. And so through this process of binding, it's sending the signal, again, that chemical signal, to that postsynaptic neuron to fire. Right? And it's the organization of these firings. I'm going to answer the phone next time I hear one ring in class. What's that?
It puts it on speaker.
Yeah, exactly. So that postsynaptic neuron receives a signal, and it's the organization of those firings of the network that is the process of kind of perception, if we're talking about the sensory system, or cognition, if we're thinking about higher order regions. Now, some neurotransmitters come out into this synapse and don't bind with the receptors here. After a period of time, if they don't bind with the receptors, they go through a process of reuptake. So if they can't find a home down here in the postsynaptic neuron, they get taken back up into the presynaptic neuron, ready to use the next time. There are some drugs that actually act to block this reuptake. And so if you know anybody that, for example, takes Zoloft. Zoloft is what we call an SSRI, or a selective serotonin reuptake inhibitor. And it's exactly as the name would suggest. If these are serotonin neurotransmitters, what Zoloft is doing is blocking that serotonin from being brought back into that presynaptic neuron. So now, all that serotonin is continuing to hang out in the synapse, waiting to then bind again with that postsynaptic neuron. That's one way drugs work. Sometimes you'll also have drugs that will stimulate the transmission of these neurotransmitters. So now, this presynaptic terminal is being told, okay, release a whole bunch more serotonin or a whole bunch more dopamine. We're going to flood the system. You also have some drugs that do neither of those, but mimic the neurotransmitter. So now you have a drug that exists in that synapse and is binding with the receptors and tricking those receptors into thinking it's the actual neurotransmitter those receptors are looking for. So I've got a slide here in just a bit that goes over some different drugs and the mechanisms by which they work. But knowing that is essentially the foundation for any sort of pharmacology that you may have interest in if you're going to medical school, or just for knowledge's sake. You're either blocking the uptake, you can also block the transmission, but block the transmission, block the reuptake, enhance the amount of transmission, or, what was the fourth one I haven't said yet? Mimic the neurotransmitters themselves. And so here are just 5 neurotransmitters. There are more than this, but these are kind of the most common neurotransmitters I'm sure you've heard of. Acetylcholine, that's how you pronounce that, glutamate, dopamine, norepinephrine, and serotonin. Different drugs play with these different transmitters, neurotransmitters. So things like Ritalin, any of these ADHD medicines, are largely targeting dopamine responses. It's why they have different effects on people who either have ADHD or don't have ADHD. Right? It's still the same stimulant, but those who do have ADHD, it serves to focus them on certain tasks, removes kind of the external distractors. Those who don't have ADHD, it serves as a stimulant to kind of wake them up and get them ready to go and focus on all sorts of these things. There's debate about whether things like Ritalin or any of these sorts of drugs help those who don't have ADHD. And if you, when they do controlled tests, they look at overall performance on, for example, recognition or recall on a test. And they find that those who don't have ADHD, it does work well for those who do have ADHD, but for those who don't have ADHD, they don't perform any better when they take the medicine, but they think they perform better because they have a flooding of dopamine that makes them feel better in the synapse. So you feel better about your crappy performance because of this release of dopamine. Which is my nerdy way of saying don't abuse those types of drugs. You'll feel better about being bad at something. So, like I said, we have things like SSRIs that prevent, in this case, serotonin from being taken back up into the presynaptic neuron. You have an increase or decrease of those neurotransmitters. And then you also have ones that mimic the neurotransmitters. And the mimicry is how things like opioids react to the brain. So things like heroin or morphine, what they're doing is introducing chemical compounds into the brain that mimic the neurotransmitters of interest. This is also one of the reasons Narcan. So Narcan is a nasal spray that helps those who have overdosed on opioids potentially have their lives saved. And one of the reasons it works so quickly, you can actually see videos online of, for example, police who administer this Narcan to people who have overdosed on heroin and how quickly they come out of it. Because what it's doing is just playing with the body's interpretation of those neurotransmitters, right? So whereas if you drink too much alcohol, you still have that alcohol in your system. The alcohol is still being processed through your system. But with the Narcan, it's able to essentially prevent that neurotransmission almost immediately. And that's why people are coming out of it so quickly. Alright, so talk to your neighbor about why it might be evolutionarily advantageous to have a nervous system which abides by this all or none approach rather than simply having a graded action potential. Talk to the neighbor and then we'll bring it all back together. Take 42 more seconds. All right, so who has a theory about why we may have evolved this way? Yeah?
If we were responding to every little thing, like because we have that threshold that you have to pass, it has to be like a certain level of stimulus. So we would go just waste energy responding to like stimuli that don't matter.
Yeah, exactly. So maybe we can think about it as one of those screens they have on a microphone, right, that attempts to essentially provide some initial blockage of pressure waves that can be registered. And so we want to filter out some of this kind of low-grade stuff so we're not just passing information down the cells. Where you get this kind of runaway effect of firing is in seizures, like grand mal seizures. So what a grand mal seizure is, it's a seizure that starts in a specific part of the brain and then moves to the rest of the brain, right? And so you have this overactivation of all these regions of the brain. One way they do that is by cutting down the middle of your brain on an area we call the corpus callosum, which is transmitting information from the left half of your brain to the right half, and vice versa. And so it's essentially, we could argue that this sort of all or nothing serves a similar purpose. We're trying to stop the activation downstream wherein we don't want to be having minute activation. Who can think of another reason?
It makes it easy for your brain to inhibit the signals at a time your activity is decreased, like when you're going to sleep.
Yeah, so a very similar idea. Again, we're going to try to prevent overactivation at a time where maybe there's not a lot of firing. You know, you're going to have sensory input most of the time throughout the day. And it's one of the reasons why when you go into like a sensory deprivation chamber, a lot of people experience hallucinations because you're still having these firings of the neurons. But by having this threshold that the neurons have to reach, maybe we're able to prevent this kind of spurious firing that could produce some sort of hallucinations. Another potential theory is that it's inherently taxing on the system to have an action potential, right? And so we want to provide some sort of barrier to entry so we're not using up a whole bunch of energy processing things we don't really need to be processing, right? And so if you are able to put a limit on the amount of times your brain is firing when it doesn't need to be firing, you're reducing its overall energy consumption and then thus making it easier to survive in the long run. Questions about anything we've talked about so far? Awesome. So as promised, here is a nice big list. Up top, we have the various neurotransmitters, their overall effects, drugs that increase or mimic that, and then drugs that decrease or block that effect. So, let's look at dopamine, for example. So, increase of dopamine leads to increased alertness, increased happiness, and decreased hunger. Right? So again, this sounds a lot like the effects you would have on various ADHD medicines. People tend to report not being all that hungry, can go the entire day without eating, without even realizing they haven't eaten. It increases that alertness, but can also increase the happiness. So drugs that are increasing or stimulating dopamine response are amphetamine, cocaine, Parkinson's drugs, like L-Dopa, and we'll show an example of that in just a minute. MAOIs, LSD, Wellbutrin. You also have drugs that attempt to block the release of dopamine. So certain antipsychotics, so when somebody's having a psychotic break, one of the things we want to do is try to prevent the release of dopamine, to cut off that sort of psychosis. Let's see, what's another good one here? So histamine. So I'm sure everybody has heard the term antihistamines, right? So if you're taking Benadryl, that's an antihistamine, or Claritin is an antihistamine. Histamine, increase of histamines in their natural form increase wakefulness, stomach acid, itchiness. Lower histamines mean lower hunger. Increase in histamines leads to lower hunger. Drugs that activate or mimic histamine production are things like opiates. And then things like Benadryl or antipsychotics or Zantac are these antihistamines. And that's why you'll sometimes see things like non-drowsy Claritin, right? Because Claritin, in its natural form, is an antihistamine. Reducing the histamine response reduces overall wakefulness, right? And so by providing that antihistamine, you've now reduced their overall wakefulness and lead them to be more sleepy. And so you can go through this. Later in the semester, we're going to talk about like psychoactive drugs. We'll talk about the different drugs and their effects on your system. But you can see here that a lot of the drugs that are abused tend to be activating these various neurotransmitters. Nicotine activates or mimics acetylcholine. You have amphetamines, cocaine, like you said. You have psychedelics on serotonin, etc. And so here's an example of L-DOPA. So Parkinson's is a really debilitating disease, especially as it continues to progress. It's taken down YouTube with it. This is annoying. There we go. All right. So Parkinson's is usually manifest in like shaking, tremors, lack of voluntary movement. So this is an actual patient with Parkinson's being asked to walk across the lawn. And you see kind of this patented shuffle step in his attempts to traverse the lawn. You'll see if you look closely, I know this is really grainy footage, but the hands, the head, the shoulders, you'll periodically see kind of shakes and tremors here. Here is him attempting to turn around on the grass, move back. And now they're going to give him a treatment of L-DOPA, which is increasing the dopamine production. And what you'll see here, in just a second, is almost a complete restoration of motor movement and motor control, right? So by increasing the dopamine production in his brain, he's able to essentially resume a normal gait there. We'll see when he goes and turns around in just a second that he has none of that little stutter step, right? It's a much more naturalistic walking behavior. One of the problems, though, with L-DOPA is that you get diminishing returns with repeated administration, and so it's not a long-term solution to Parkinson's. It's a good temporary remedy for Parkinson's, and that you can give this to the person. The effects will wear off shortly, but it doesn't help essentially mollify the long-term effects of Parkinson's, nor does it slow the degradation of the nervous system. So now in the last 10 minutes, we're going to talk about your nervous system as a whole, because that's just what we're going to do, even though it's a lot more important and more nuanced than what we're going to talk about. We can break it up into peripheral nervous system and then your central nervous system. Right? So you may have heard CNS, central nervous system, and PNS, peripheral nervous system. The central nervous system is what we're referring to as your spinal cord and your brain. Right? Just think about everything central to your body. Right down the middle. Your peripheral nervous system we break into somatic and autonomic. So somatic controls the voluntary movements of skeletal muscles. So any movement you're making, you're engaging your somatic nervous system. Peripheral is that the autonomic is controlling the self-regulated actions of internal organs, glands, so things like digestion, your heart rate. Breaking it up even further, we can split that autonomic, or think automatic when you think autonomic, nervous system into two different responses, sympathetic and parasympathetic. So sympathetic is that kind of heightened arousal. So think before you do something, when you're really nervous, kind of your heart rate elevates, you've engaged your sympathetic nervous system. The parasympathetic is that kind of calming response. So the central nervous system is responsible for integrating the sensory information and responding accordingly. So it's getting all sorts of information from the body. You're getting information about proprioception. So what is my body's orientation in the environment? Getting information about pain. And then, depending on what sort of information and from where it's coming, it either triggers an automatic response, it innervates with efferent neurons, which are going to your muscles to trigger some sort of response. Right? And this is what happens when you go to the doctor and they hit your, whatever the tendon that is, patella tendon, I think, in the front of your knee. Right? They're trying to activate that response. And then you also have that information going up to the brain for a higher order processing. It's what allows you to feel cold on a day like today. It's being processed at those higher levels. So again, you have the somatic, you have the autonomic. The somatic is the voluntary control over muscles. So if I ask you what system is responsible for voluntary control, your answer would not be peripheral nervous system. Your answer would be somatic. Right? Because peripheral also contains those autonomic components. And so think about the autonomic engaging any parts of your body you don't have voluntary control over. Gland secretion, digestion, respiration. I mean, you can have voluntary control over respiration, but it's not something you have to focus on. So the sympathetic nervous system is what you can think of as fight or flight. Right? I'm sure you all have heard this before. It's the idea that when activated, you tend to have two responses. It's either fight in that moment, fight off whatever that threat is, or flee, run away. Over the last decade or two, we've started to understand it's a little bit more nuanced than that. Fight or flight tends to predominantly be the masculine response. Women tend to have what we call, because we have to make it rhyme, tend and befriend. Right? And so wherein there's a stressor, women tend to more frequently try to take care of others. They also try to surround themselves with more people, more like-minded, more sympathetic individuals. It's why I'm sure in your social life you've seen, particularly in relationships, when there's conflict in the relationship, guys tend to withdraw. Women tend to talk it out with other people. Extreme activation of the sympathetic nervous system can lead to essentially an overstimulation of the system where we've lost fine motor skills at really high levels. We can even lose gross motor skills, so big movements. You can think about this. You know, if on Halloween somebody jumps out and scares you as you're getting ready to put your key in the door, you can't put your key in the door anymore, right? You've lost all fine motor skills, the ability to manipulate on a very small level. It's also why before maybe a big interview or a big game, you'll feel nauseous, upset stomach, ready to vacate the fluids. Most fascinating to me is the fact that a lot of folks in combat have this sort of response, right? Cover your ears if you're offended by language, but what it's colloquially referred to is battle shits, right? So men and women will actually vacate their bowels because of this overstimulation of the sympathetic nervous system. The parasympathetic response can sometimes be a backlash to that sympathetic response. It's why after a period of really high anxiety, you feel exhausted, completely depleted. That's your parasympathetic system kicking in and regaining control over the functions that were essentially abandoned during that sympathetic response. Right? During the sympathetic response, your body says, we don't have to worry about digestion now. We need to get the heck out of here so we're not being chased by a tiger. After that, though, once your heart rate decreases, once your respiration and blood pressure decrease, your parasympathetic system kicks back in and says, all right, we're going to take back over control of digestion now. And again, because of my interest and where I came from, there are a lot of really interesting case studies of these sort of parasympathetic backlashes in combat, wherein the most vulnerable time for a military is right after some sort of skirmish or engagement. That's why a lot of armies will attack immediately thereafter, because the other army is experiencing this sort of parasympathetic backlash. They're exhausted. They're not being aware of their surroundings. And there are actually cases of people legitimately just falling asleep in combat because of this parasympathetic backlash. Any questions from what we covered today? None? Cool. Have a good weekend. I will see y'all on Tuesday.